Hello, everybody, and thank you for joining us today at our Precisionary Webinar series. And it's my pleasure to introduce Dr. Joannah Fergusson, or Jo, who is a scientific officer at the Kennedy Institute and the NIHR Biomedical Research Centre at the University of Oxford. Dr. Fergusson has dedicated her career to human immunology and developing in vitro models of human immunity. Her current research focuses on understanding the mechanisms of various vaccine modalities in order to design more effective vaccines against infectious diseases, including SARS-CoV-2. Her work aims to identify early inflammatory events in human lymph nodes that influence protective immunity and extend this research to other tissues in chronic inflammatory diseases. And today, Dr. Fergusson will discuss the development of precision-cut human lymph node slices and state-of-the-art techniques like single-cell RNA sequencing in imaging to model adjuvant-induced inflammation. She will also address challenges in developing new vaccine modalities. And we all know this with the recent COVID pandemic and vaccine development. She's going to introduce precision-cut lymph node slicing as a model for vaccine studies and describe the mechanism of an open-source adjuvant from human lymph node studies. So please join me in welcoming Dr. Joannah Fergusson. Thank you so much, and it's our pleasure to have you today.
Thank you, and thank you for the opportunity to present some of my work here today. So yeah, I'm going to tell you a bit about precision cutting of human lymph nodes and using this as a model to investigate vaccine adjuvant mechanisms of action. So the impact that vaccines can have on global health has become really starkly evident in the last few years. But even COVID aside, the benefit of vaccines to global health is really massive, and it's been estimated that that vaccines in general have averted around 50 million deaths, and this is across a range of different infectious diseases for which we have effective vaccines. But there are still many different challenges still in vaccine development, and I'm just going to highlight a few here. So one of these is in vaccine coverage. So while we may actually have vaccines that we can use against different infectious diseases, getting these vaccines to the right people and the right places still remains a challenge. And so this is data from the World Health Organization. And you can see that while in much of the Western world we're getting really high levels of vaccination, there's still these pockets where vaccination is low. And while the percentage coverage may look like fairly okay, this actually correlates to large numbers of people. So 40 million people, 40 million children did not receive an initial dose of this diphtheria, tetanus, pertussis vaccine. And then a further 6.2 million are only partially vaccinated. And this relates to the fact that, as you can see here from this list, these vaccines, many of these vaccines require multiple doses to enable the full level of protection. And so while you may be able to vaccinate with one dose, especially where people have difficulty in accessing health services and immunization sites, it becomes quite difficult to reach that level of full protection when they have to come back for multiple doses. There's also, of course, emerging diseases, and we all became aware of this in particular with COVID, but these are other diseases, infectious diseases, that the World Health Organization has highlighted as ones of to kind of be concentrating on in terms of research, while they also acknowledge that there may be another disease which we are not yet prepared or aware of to that degree, which may in the future become a global health concern. And then there's also the difficulty or the challenge of making sure that we can vaccinate all populations. So older people have reduced responses to vaccines. They suffer from this condition called immunosenescence, where their immune populations become less responsive. There's also a general heightened inflammation. And then also our younger populations as well, so vaccinating children and ensuring that we can vaccinate these populations safely into a high level of efficacy. And then there's also those who suffer from chronic inflammation, other comorbidities, that may also impact their response to the same vaccines. So if you were looking to design an ideal vaccine, you would want to have one that could induce protective immunity, and this would need to be a kind of broad range of immune responses from cell-mediated immunity to antibodies, and you would want this immunity to be long-lasting, so to give you protective immunity throughout life, ideally. And you'd want to be able to induce this with only a few doses, one ideally, and those doses to be low to reduce the burden of manufacturing these vaccines. And you would want these to work in all populations from young to old. And it's here that adjuvants can really help. By adding an adjuvant to your vaccine, adjuvants can act to broaden immune responses. They induce T cell responses, which can impact the antibody production. They have the ability to increase durability and allow smaller doses of the vaccine to be used, which enables dose sparing. And they also have the potential to overcome immunosenescence. So adjuvants are broadly defined as substances which, used in combination with a specific antigen, produce a more robust immune response than if you use the antigen alone. And this was described back when they were first identified more than a century ago. And this was by Ramon, who found that when he vaccinated horses, they had a more robust immune response if they also developed inflammation around the site of injection. So broadly, adjuvants work by inducing inflammation, which kind of heightens the immune response. But for much of the following century, there was only really one adjuvant which was licensed for human use, and this was alum salts. And so the development of adjuvants has also been described as one of the slowest processes in the history of medicine. Now we do have a range of different adjuvants which are used in a range of different vaccines, and this of course has also been aided by the pandemic and the kind of fresh impetus to kind of design vaccines. But in general, our understanding of the mechanisms of action of these adjuvants is still fairly poor. And this was from a recent review which talked about some of the challenges in adjuvant development. And when we have novel adjuvant candidates, we test these in preclinical models and evaluate the immunogenicity and some safety parameters. And then these then progress into humans, which involves the use of clinical trials, which are obviously very costly. And it's from then they go through safety, immunogenicity, and efficacy trials. And if they succeed, then that's great, they can be licensed. But if they fail, then we're not necessarily very clear why they have failed. So in the lab, Soren in the lab has looked at the mechanism of action of certain vaccine adjuvants. And to aid the study of vaccine adjuvant mechanism of action, the Vaccine Formulation Institute, based in Switzerland, has developed open source adjuvants, which can be used clinically, but also are globally available for also these mechanism of action studies. So Soren had access to 4 different adjuvants. So these are either liposomal based or emulsions, and then they all contained this saponin, which is called QS21, and then with or without a TLR4 agonist, which was MPL-like. And just to emphasize the importance of adjuvants and their different mechanisms of action, so Soren tested this in a mouse model of malaria. So he gave the mice a priming vaccine, followed 3 weeks later by a boost, and then challenged them with malaria. And this was together with the R21 malaria vaccine. So the R21 malaria vaccine alone, all mice died 6 days post-challenge. But with the addition of adjuvant, you saw improved protection of the vaccine. But there was a hierarchy of response of the different vaccine adjuvants. So there was a high level of protection with the LMQ and SQ adjuvants, with lower levels of the SMQ and LQ. And this also showed us that it's not just the presence of the TLR4 agonist that makes a difference, as SMQ, which also has a TLR4 agonist, performed more poorly than SQ, which did not. So Soren did a lot of work to look into the mechanism of action of protection of these adjuvants. So interestingly, all 4 adjuvants induce similar levels of antibody, total IgG, and yet the ability of these to inhibit invasion of malaria parasites into hepatocytes was lower in LQ, but similar between the 2 protective adjuvants. So trying to look into what may be different between these different protective adjuvants, the LMQ and SQ, you could see, observe that although the total IgG levels were the same, there was a difference in the composition of these adjuvants with IgG1, IgG2A, B, and 3. And this correlated with a different T helper index, which then impacts the different isotypes of antibodies that are induced. With the LMQ inducing a higher T helper 1 type response with interferon gamma and some IL-13, which is a T helper 2, whereas the SQ was higher in T helper 2 type responses. And this really resulted from the earlier innate responses to these different vaccine adjuvants. So with LMQ, he looked at cytokine production within the site of injection, so the muscle, and also within the serum. And LMQ induced large numbers of different cytokines, whereas the cytokine profile of SQ was much lower. And importantly, Soren was able to identify that the mechanism of action of LMQ was reliant on the NLRP3 inflammasome. So if you took mice that had a working NLRP3 inflammasome or a knockout for NLRP3 and took bone marrow derived macrophages and treated them with the different adjuvants, you could see that the IL-1 beta production of LMQ, so LMQ induced it. IL-1 beta, but this was knocked out when you also knocked out the NLRP3. So just a note on the NLRP3 inflammasome. So this is the NLRP3 inflammasome activates caspase 1, and this allows release of cytokines IL-1 beta and IL-18 from the cell, but it also requires a signal 1, which comes through TLR4, for instance, or other pathogen-associated molecular pathogens. And this increases the transcription of the proforms of these cytokines, so that when the inflammasome is activated by the second signal, so in this case the QS21 part of the adjuvant is activated, and there are high transcripts of IL-1 beta and IL-18 which can then be cleaved into their active form. So through this, Soren identified that LMQ and SQ had different innate and early responses, but they actually converge on protection from malaria, even though they're via 2 different mechanisms of action. So while these studies are really useful in enabling us to really understand different mechanisms of action, and of course, many of us will be aware of the pros of using animal models, because they enable us to actually test in vivo. We can have access to the different tissues of interest, and we can also genetically manipulate, such as the knocking out NLRP3. But there are also species differences in terms of the cell populations that may be present in these animal models compared to humans, the receptors they express, and also the mediators that they express. And just one illustration of this was Eloise, a PhD student in the lab, took published data sets of single-cell RNA sequencing, so the transcriptome of different populations from the mouse lymph node and the human lymph node. And thinking about that TLR4 agonist part of the adjuvant, she looked at those receptors for the TLR4 agonist, which may be present across the different populations. And whereas in mouse, one of the major cell populations expressing TLR4 were these lymphatic endothelial cells, if you look within human, there's actually very low levels of expression of TLR4 and the associated co-receptors. In mice as well, the neutrophils expressed higher levels. There was some expression within the human neutrophils, but it was really the monocytes which showed the higher level of expression. And the impact of this is that results often then don't translate between what we see in the animal models and what we observe in humans. And this can be part of the problem then with when something fails, knowing why it's failed, when in our preclinical animal models they may have shown efficacy. So for this reason, people are trying to turn to models of human immunity to complement these animal studies. And this is needed to try and improve this pipeline of getting drug candidates or adjuvants, vaccines through the pipeline. And while there are many different, very elegant approaches to doing this, many of these approaches rely on trying to recapitulate native tissue architecture and to try and reform them from their constituent parts. But what the approach that we wanted to take was to find a model where we could use relevant human tissue in a way that would enable it to be cultured so that it retains the native tissue architecture, so that you can retain those native cell-cell interactions that come from cells being in proximity. But it would need to be able to be cultured in a way that would also be susceptible to perturbation to allow us to add our drugs or adjuvants of interest. And then this would enable us to study the mechanism of action, but within the right context of human tissue. So to this end, I have been working to develop the lymph node culture system, and we turn to precision cutting of lymph node slices. So cutting of lymph nodes proved very challenging in the first instance. So we had access to a vibratome, and it was near impossible to get slices of lymph nodes from this machine. But then we had access to the compressotome, and we brought one into the department. And it's here that the ability to precision cut lymph nodes really became possible. But obviously, human lymph nodes are a rare source. So to do our kind of preliminary setup of the model, we used pig lymph nodes, which were more readily available and actually share 80% immune parameters with humans. So they're not a bad model, actually. So we were able to get pig lymph nodes, and with the compressotome, cut these into slices of different thicknesses. And when we cultured these slices of either 300 or 400 micron thick, these retain viability much better than a whole lymph node, which very quickly within the first hour or so became hypoxic, and certainly after overnight culture was completely dead. I performed some optimization of the culture conditions, so different volumes, and then adding in our adjuvant and ensuring that viability was retained. So you have the viability of the cells within the slice, but also the during culture, some escape into the medium. So you can also look at those cells. And importantly, our slices remained responsive to the adjuvant. So here, there was a dose-dependent response of the slices to adjuvant with the transcription of different inflammatory cytokines, so IL-1 beta and IL-6. We then wanted to perform some in vivo versus ex vivo comparisons, so turned back to the mouse model for this, where we could do both sides of that comparison. So firstly, to test that we could get back our cells of interest, so comparing cell populations from a lymph node taken directly versus our ex vivo slices. And this is after culture. So here then we can get back our different innate cell populations. So we've got dendritic cells, neutrophils, monocytes, and macrophages still viable at the end of culture. And then by using a fluorescently labeled liposome, a fluorescently labeled LMQ adjuvant, we could then track uptake of the adjuvant across these different cell populations. So either after intramuscular injection into the mouse and then looking at the draining lymph node, this is the in vivo condition at 3 hours, or later at 24 hours, compared to our slices where the adjuvant has been added to the culture. And when we look here, we see a similar hierarchy of uptake with the same cell populations taking up the adjuvant ex vivo in the slices as in vivo in the draining lymph node. And generally, the uptake increased over time with the higher levels of uptake within the 20-hour time point. And so that's the time point we took forward to try and increase our ability to see the maximum impact on the different cell populations. We were also able to use this fluorescent adjuvant to look spatially where it drains to within the draining lymph node. So this is after intramuscular injection. We can see that it pulls around the subcapsular sinus, but also flows into the medullary region of the lymph node. And here, if we take our slices, we can also see that there is adjuvant uptake around the, or pooling around the outside subcapsular sinus, but also within the medulla. And this was also an active process. As we fixed our slices before culture, there was no evidence of any adjuvant, whereas those that were unfixed had still taken up the adjuvant to enable imaging. So having optimized and convinced ourselves that this was a fairly good model of what was happening in vivo, we then went on to perform our human studies. So we get human lymph nodes from gallbladder removal surgery. So these the gallbladders are generally removed for conditions such as gallstones. They're removed at the point of being non-inflamed, and the surgeon inspects for inflammation, and so we get the non-inflamed lymph node that is associated with that gallbladder. So when the gallbladder is removed, there is often a lymph node associated with it, which would usually just be disposed of, but instead we can get and use in our studies. So we take these human lymph nodes, and with the help of the compressome, and I think here it's really the point of compression which helps us to cut this, which is with the compressome technology. This compression allows us to cut through the fairly elastic tissue of the lymph node and enable us to get these 300 micron thick tissue cross sections. But the human lymph nodes, as you can probably see from the picture, they were quite different from the pig ones that we had been using, and they do vary quite significantly from donor to donor. And we were having trouble in actually cutting these lymph nodes, and particularly cutting serial sections. So we found this paper from a group at the University of Virginia who had been precision cutting mouse lymph nodes, and found a difficulty in cutting those lymph nodes which had come from mice which had been vaccinated. And they had incorporated a mild detergent wash of the lymph node to remove surrounding lipid droplets. When you've taken the lymph node out of the surrounding fat, there's still some adipose tissue or lipids associated with the lymph node. So by doing a mild detergent wash, this removes that and enables it to be embedded better into the aggros, which is required for cutting. And so just to check that this had no impact on our downstream viability, we check that those which have been cut with or without this digitonin, which is the mild detergent wash, retain viability. And at the thicknesses that we were using, so 300 micron mainly, we could retain viability even after this detergent wash. And so this then enabled us to increase our frequency of getting serial sections from the same lymph node, which enables us then to do paired tests of control and treated slices within the same donor. So we then also wanted to do a similar check for our cell populations of interest within the human lymph nodes compared to a whole node. So we take a whole lymph node, digest it directly, and stain for different cell populations, or a slice that's been cultured, and then also stain for the same cell populations. And the majority, or the main populations within a lymph node, are the lymphocytes. They're 95% of the cells you'll find there. But beyond these cells, you also can detect dendritic cells and macrophages, and we could harvest these cells from slices as well as the whole node, even neutrophils. And then in addition to hematopoietic cells, there are also the stromal non-hematopoietic element, and we could also harvest back the various populations of these, including fibroblast reticular cells, lymphatic endothelial cells, and blood endothelial cells. And although we can't do the same kind of comparisons of adjuvant uptake of in vivo versus ex vivo, obviously in the human setting, we could do, we added LMQ, the fluorescently labeled LMQ, to our culture medium and tracked uptake, and did see that there was a hierarchy of uptake across the different cell populations, with lymphocytes and neutrophils showing low-level uptake, whereas our myeloid cell populations, and also interestingly our stroma cell populations, also showed uptake of the adjuvant. So with this, we then had a model by which we could take a lymph node, precision cut slices and have these paired conditions of control or treated slices. We could then digest these slices into single cell suspensions and analyze this by flow cytometry. So that's the data that I've just shown. But we also wanted to check that we were really retaining the cell-cell interactions in the tissue architecture within the slices. So we took slices after culture, cut them down further into 5 micron thick, paraffin embedded and fixed sections onto microscopic slides, and then took this forward to hyperplex imaging. So this is with the cell dive system, whereby you do multiple rounds of fluorescent antibody staining, bleach these antibodies, and then do the next round to build up a panel of markers. And with this, we could see that the characteristic morphology of lymph nodes was retained in these slices. So we have still our B cell follicles, which are intact, our T cell zones, and our antigen-presenting cell-rich medullary sinuses. And then what's really nice about this system too is that we can also look within the supernatant at what inflammatory mediators have been produced and released into the supernatant. So the supernatant cytokine analysis after treatment with our LMQ adjuvant, we can see the production of various different inflammatory cytokines. Although you can also observe here that there is a wide donor-to-donor variation in the type and the amount of cytokine which is produced. But because, as I mentioned, these are paired conditions, we can look at control versus LMQ treated and observe whether there are significant increases in cytokine. And indeed, we see a significant increase in TNF alpha and IL-6 upon LMQ treatment of the slices. So with this, we had our model for mechanism of action studies in humans, which is this culture system which enables to retain tissue architecture, but can be cultured. We can add in adjuvant and see this dose-dependent response to the adjuvant and induction of inflammatory cytokines. So with this, we could then look at a mechanism of action in the right context of our human tissue. So to move on to some of the results we've had from these studies. So just as a reminder, we concentrated our studies on this adjuvant LMQ, which had shown the highest level of protection against malaria. And as a reminder, this is the liposomal adjuvant with the saponin QS21 and the TLR4 agonist. So with this, we took our healthy human lymph nodes, cut them into 300 micron tissue slices. We cultured these separately, but then pooled multiple slices together to account for differences in the point at which you may have cut, but then corresponding to differences in cell populations that you might get back. So we took our slices, had control or adjuvant treated conditions, and then dissociated these for single cell transcriptomic analysis, so 10x chromium. We discarded the red blood cells and dead cells, and also because, as I mentioned before, lymphocytes are the majority of the cell types within the lymph node, we didn't want this signal to dilute out any other interesting signals from the rarer cell types. Therefore, we sorted out the majority of T and B cells and then spiked them back in at a defined ratio of cells so that we could also get a flavour of what was happening in these populations. So with this, we were then able to generate a map of our different cell populations, so including different lymphocytes and stromal cell populations across 6 different donors, control and treated. And this enables us then to look on a population by population level at what are the gene changes that occur when you treat with our adjuvant. And just a reminder, because we've retained the tissue architecture, we also would hope to be able to see those indirect effects which occur from the cell-cell interactions beyond just the direct effects of the adjuvant itself. So to begin with, we wanted to check at a transcriptomic level that our slices were replicating those of a whole lymph node. So we also had our whole lymph nodes, which we digested directly and identified the same cell populations from these as we had within the slices. And then we performed a correlation compared the transcriptomes of those populations from the whole lymph node compared to the same populations within the slice, and taking the median gene expression of the top 2,000 genes, we could then see whether these genes were correlated in their expression between those populations. So by Pearson correlation coefficient, we can see that there is a correlation in the transcriptomes of the different populations from our slices to our whole, with the exception of here the granulocytes. So our neutrophil granulocytes did not match from our slices to the whole, and therefore were excluded from any future analysis. So when we were looking to see what cells may be initiating our inflammatory response to LMQ, we looked to see expression levels of the receptors for the TLR4 agonist part of the adjuvant and its downstream mediators, and for the NLRP3 inflammasome. And when we looked here, we could observe that our monocyte macrophage cluster had expression of TLR4 and the co-receptors which are required for signaling, as well as some expression of all the downstream components, and also expression of components of the NLRP3 inflammasome. So if we look here at the genes which are differentially expressed in our monocyte macrophages when we've treated them with the LMQ adjuvant, and then look for those pathways which are upregulated in those genes, we can see that there is an enrichment in this pathway of toll-like receptor signaling, which suggests that there is TLR4 signaling within our monocyte macrophage population. And also, if we look at gene signature for the inflammasome, so here we look at the expression of the inflammasome genes across our different cell populations and see if this changes upon LMQ treatment, we can see a significant upregulation in this inflammasome gene signature within the monocyte macrophage population. So this together suggests that indeed the TLR4 and the QS21 of the LMQ are synergizing to activate these macrophages. And when we look within the cell supernatant, we can indeed see these inflammasome-mediated cytokines. So as I mentioned before, when the NLRP3 inflammasome is activated, it results in the release of these cytokines IL-1 beta and IL-18, and these are found at elevated levels within our treated supernatants. And what's really nice is that we can also interrogate the pathways of stimulation through pre-treating our slices with molecular inhibitors of these pathways. So we have an inhibitor of the NLRP3 inflammasome here, this MCC 950, and also of TLR4 signaling with the TAC242. And we can see here that both of these inhibitors mediate a reduction in IL-1 beta release, whereas with IL-18, the inhibition of the inflammasome resulted in a reduction of IL-18, which was not so reliant on TLR4 signaling, which suggests that this IL-18 is not as reliant on this signal 1 of the inflammasome, where you needed upregulated transcription, but actually within cells, the IL-18 transcript is ready. So when the inflammasome is activated, it can cleave the IL-18 for release. So knowing then that we have induced release of these cytokines from the monocyte macrophage population, we then look to see what the downstream effects of that may be. Now, IL-1 beta, when combined with IL-23, is an activator of ILC3s. So these are innate-like lymphocytes, innate lymphoid cells, which can be grouped into different groups depending on their transcription factor expression and the activating cytokines and the cytokines they release. One such group of these, the group 3, are activated by IL-1 beta and IL-23 to release either IL-17 or IL-22. And these innate lymphoid cells can change the microenvironments and the environments within tissues due to their sensing of cytokines and their release of other cytokines. And when we looked in our whole and indeed our slice populations, with the majority of the cells actually within the lymph node are these ILC3 populations, which was it was a surprise to us just how big a proportion they were. And we do indeed see IL-23 also induced by LMQ within the slice culture supernatants. In addition to this, we also see the appearance of an activated subset of ILC3s. So while these appear upon slicing, we then see an increase in this population, this activated population, when we've added LMQ, and this is concomitant with a reduction in the NKp44 ILC3 population, suggesting that this is the population which is becoming activated to produce IL-22. So as I mentioned, when ILC3s are exposed to IL-1 beta and IL-23, they produce either IL-17A or IL-22. So we didn't see any real production of IL-17A in our culture supernatants, but we do see some suggestion of IL-2 and T2 production upon LMQ treatment, although this isn't true of every donor, again showing that this retains the kind of donor-to-donor variation, but was only seen in 3 out of our 4 donors tested. And then we've also got this IL-18 production, and IL-18 can, in combination with other innate cytokines, particularly IL-12, can activate the group 1 ILCs, which includes NK cells. And again, these populations are a major population within our lymph nodes, both NK cells, which are the 2 green populations, and ILC1s. And while we don't see any evidence of IL-12 within our culture supernatants, we did see a robust upregulation of interferon gamma within our NK cell subsets. And this was also detected within the culture supernatants, suggesting that the NK cells really are activated by this IL-18, potentially in combination with other cytokines such as IL-2 or IL-15, to induce production of interferon gamma by the NK cells. And interestingly, we could also see that inhibiting the inflammasome and TLR4 could inhibit the production of interferon gamma. And while NK cells hadn't shown evidence of TLR4 expression or NLRP3 inflammasome at baseline in those dot plots I showed earlier, this suggests that the upstream inhibition of these cytokines could have a knock-on effect to the ability to produce interferon gamma. So with this, we have a model by which we can see the direct effects of the adjuvant on myeloid cells, and then the downstream indirect effects on innate lymphoid cells, including these NK cells. And because we can look spatially within the lymph node as well, we wanted to confirm that this was likely to be true of the cell populations if they were in proximity to each other. So we wanted to test where the NK cells were. So by staining for CD3 negative and CD56 positive cells, we could identify our NK cells. And indeed, they were neighboring our CD68 positive macrophages. But we also observed that there were B cells nearby. And although this is outside of the B cell follicles, there were areas of extrafollicular B cells which were in proximity to these NK cell macrophage pairs. So we wanted to look at the impact on B cells, which would then have a kind of potential impact on our protective immune responses and antibody production. So as I'd said before, we depleted the majority of B cells, but we had spiked in a population here. And when we looked at the genes that were upregulated within these B cells upon adjuvant stimulation, and then looked for enrichment of different gene pathways, we see enrichment of interferon signaling pathway, which again suggests that the NK cell interferon is signaling to these B cells, and this results in an activation of the B cells suggested by the upregulation of these pathways, including metabolism of RNA, but potentially more interestingly, this antigen processing presentation pathway, suggesting that that the adjuvant could downstream through NK cells result in an increased antigen processing and presentation within B cells. And then also to go back to what I had mentioned earlier about the differences between mouse and human and within the lymphatic endothelial cells, the advantage of this using precision cut slices is that we can retain these populations, these stromal populations of interest. So this was both when we digested down single cell suspension, but also we can stain for these populations by protoplanin expression and CD31 for our endothelial cells. So when we again look back at these dot plots of expressions of our mediators of the LMQ activation and look at our stromal cell populations, we do see that although it's a low-level expression, there is expression of the different receptors and co-receptors of TLR4 within the endothelial cell population, and also some low-level expression of NLRP3 inflammasome components. And so when we again look at the genes that are differentially expressed between control and LMQ adjuvant treated stromal and endothelial cells, we see that within the endothelial cell compartment, we again see an enrichment of toll-like receptor signaling, suggesting that the TLR4 agonist is directly signaling to the endothelial cells, but also in both populations we see an enrichment in IL-1 signaling, which suggesting that the IL-1 beta produced mainly by monocyte macrophages is also having a feed-forward loop to these stromal and endothelial cell populations. And then we also see an enrichment in pro-inflammatory and pro-fibrotic mediator pathway within both of these cell populations. And within this, it includes a variety of different chemokines, including CXCL2 and CCL5. And while the precision cut slices is a static system, meaning that we can't look at cell recruitment within this model, so we return to our mouse model and again injected our adjuvant into the mice and then looked at various time points in the draining lymph node to look at our cell populations and how these may change over time. And when we did this, we saw that monocytes and neutrophils were rapidly recruited, in particular neutrophils, to the lymph nodes, whereas dendritic cells were also recruited, but at later time points. And this suggests that the LMQ signaling to stromal cell populations is involved in this early recruitment of innate cells into the draining lymph nodes. And interestingly, we also saw within the mouse that these fibroblasts and endothelial cells, or lymphatic endothelial cells in particular, were capable of taking up LMQ when we used a fluorescent adjuvant to map uptake. So by this, we start to build a picture of some of the mechanisms of action of our adjuvant within humans. So we have direct activation of myeloid cells in our macrophage population by the LMQ, both the TLR4 agonist and the QS21, which activates the NLRP3 inflammasome. So we have production of inflammatory mediators, including IL-6, TNF-alpha, and then the inflammasome-mediated IL-1 beta and IL-18. And this IL-1 beta, together with IL-23, can activate IL-C3s to produce IL-22. And then also IL-18, in combination with other innate cytokines, can activate NK cells to produce interferon gamma, and this then signals to nearby B cells to have an impact on their antigen presentation. We also have direct activation of endothelial cells by TLR4, with also feed-forward signaling by the IL-1 beta to both the stromal cells and the endothelial cells. And these produce a range of different chemokines that then recruit other innate cells, which may or may not be also carrying adjuvant from the site of injection. And the result of this is an overall amplification of the inflammatory response and this state of inflammation, which is known with adjuvants, which can then improve our ongoing or our resultant immune response. So that's the work we've done in mapping mechanisms of action within humans, but we're also working to try and better understand our models, our preclinical models within this pipeline of taking vaccines or drugs of interest from the bench to the clinic. So to this end, we're also performing studies in non-human primates, so we have access to lymph nodes from rhesus macaques, and we're doing similar studies here whereby we're looking just directly at these lymph nodes, at what cell populations are present, and then also by using the compressed, cutting these into slices to perform similar studies with the adjuvant, meaning that we can have data sets which are treated in the same way between humans and primates, and can look at the different pathways between these species. And to this end, we're also building up a panel of cross-reactive antibodies to enable us to also perform spatial analysis using the cell dive system of these primate lymph nodes. And with this, through improving our understanding of animal models and using a system by which we can look at perturbation within this by using precision cut slices, and also by introducing precision cut slices of human lymph nodes to model the immunogenicity and mechanism of action, we hope to provide further pieces of the puzzle, which will enable us to improve our understanding of adjuvants, and therefore their progression into the clinic, and to ensure that those that progress are more likely to be successful. And so with that, I'd just like to thank the different groups that have been involved, so those at the Kennedy and also at the Jenner Institute. The adjuvants were made available by the Vaccine Formulation Institute, and the work was funded by the Chan Zuckerberg Initiative. And thank you very much to Precisionary for the compressed, which really enabled this study in general, and for the opportunity to present. Thank you.
Oh my gosh, thank you, Dr. Fergusson. It's a pleasure having you. So what I want to do now is open up the floor to our attendees. We have a small intimate group, so please don't feel shy about unmuting yourselves or showing your video. If you would prefer, you can also enter your questions in the chat box. I'm happy to also ask it for you. Does anybody have any questions or comments for Dr. Fergusson's talk? Takes a little while sometimes for somebody to unmute.
It's a lot of information.
Hi.
I have a more technical question. Can you estimate, and I may have missed it, the amount of dose of QS21 that you're injecting in the mice?
Yes, we can. I don't have the number off the top of my head, but it's in the paper. We can estimate, the amount per adjuvant particle or liposome, and then we have the volume of adjuvant that we're adding to the mice. So we can estimate that, yeah.
Because we had some negative side effects with QS21, but although I assume we may have overshot with the dose that we gave these mice, and since we are not immunologists, we may have done too much of the good thing there for them.
I think with incorporating them into the liposome, I don't know in what format you were giving QS21, but it can be really lytic, and without the liposome incorporates some cholesterol, which helps to counteract that. So in the liposomal formulation, we see a reduction in that kind of lytic effect of QS21.
Yeah, our QS21 was mixed with a nanoparticle solution, and we don't even have a detailed characterization of what the interaction would look like.
Yeah. If you look within the Cell Reports Medicine paper, that there should be the details of the adjuvant dose in there.
All right, thank you.
Thank you.
Wonderful. Thank you for your question. I want to open the floor up to anybody else. While we're waiting, Dr. Fergusson, I actually had a little question, and of course it pertains to precision cut lymph node sectioning, because I'm with Precisionary. When you put them in culture, are there any tips and tricks for the specific types of buffers that we use to help their viability?
Yeah, so we use a complete RPMI. So that includes L-glutamine, HEPs, beta-mercaptoethanol, and FBS, and we include some penstrep in there as well, in case of any outgrowth of kind of bacteria, because obviously the lymph nodes aren't sterile from the point that we get them, although we then try and retain sterility. So yeah, we've found a complete RPMI as retaining the viability sufficiently. We plan to try and extend our culture times out further, and in which case we might need to do some fine tuning of that, but at least in the first instance, the complete RPMI works well.
Great. And you had mentioned in the beginning that it is difficult. We were chatting about cutting live lymph node tissue, right? What were some of the challenges you've had, perhaps using other vibratomes or microtomes in cutting, small lymph nodes? And how did the compressed help you resolve that issue? Like, was it gluing? Was it embedding? Tell us a little bit about
Yeah.
that process.
Yeah, I faffed around ages trying to get these things to
Uh-huh.
work. But the problem with the vibratome is, or in general, is that we had the lymph nodes are really quite elastic, so they pop out of the agros when you try and embed it. So yeah, just or the blade itself would kind of skim off the top of the tissue.
Right. It kind of bounces off when it's cutting.
Bounces off, and then eventually your whole tissue would just fall out.
Mhm.
So when I began using the compressed-ome, I think it was that compression that then really made the difference in being able to cut. We've also used, we don't use low melting point agros, we use normal agros that has a bit more rigidity. So we then have to be careful with temperature of the embedding, but that's really made a difference as well in holding the tissue in place so that we can get these sections. There's still difficulty because, especially with the human lymph nodes, it varies donor to donor. I guess how fibrotic or not the tissue may be. There's sometimes some red blood cell contamination. So it can vary donor to donor, but in general now we've got a kind of setup where we can cut these lymph nodes, and yeah, we don't glue them actually in the end because they're so small that we would never get to the end of the tissue. So we just embed them in the agros, kind of within the middle of it, so we can get through the whole tissue.
Very nice. Thank you for sharing that. You make it sound easy, although I know that it's been quite a journey to try to get it perfected.
Yes, but I think we're there now. So yeah, we're happy to share it with others.
So I just want to have one last call for any questions or comments. If not, I'm gonna say thank you one more time, Joe, for your time here and for all of our attendees. And thank you for attending. Have a lovely rest of the week.
Thank you.